Can you see my screen? Yes. Okay.
Perfect.
Okay. So thank you very much, Maria. So my name is Alessio Buccino. I'm a postdoc at ETH at Andreas Hirnemann Lab, the bioengineering laboratory. And the work that I'll be presenting today is a collaboration with the Bluebrain project at EPFL. And it's about a multi-modal fitting procedure to construct biophysically detailed multi-compartment neural models. So first of all, I would like you to quickly introduce these multi-compartment models. Actually, Carol also had some of these models, but basically these kind of models are biophysically realistic representations of single neurons, and they're mainly built of two different parts. The first part is the morphology, which is usually extracted from fluorescence images of single neurons, and we can get a very nice and fine 3D representations of single cells. And the second part, which is harder to basically construct, are the electrical properties. And these are usually fitted to electrophysiological recordings, mainly from patch clamp data, and they are used to fit the many parameters that these models are made of. And I just want to mention that in the last 5 years or so, maybe 10 years, there's been a huge international effort in building and sharing these detailed and complex models, especially from the Bluebrain project, where they have thousands of cell models from rest, sensory motor cortex, and also hippocampus, and also from the Allen Institute of Brain Science, where they contribute several hundreds of models from mice and even from human tissue. These models are usually built from signals from somatic patch clamp. So basically you apply a stimulus, you get a response, and then you try to find a model that fits your responses. But the main problem that I see here is that neurons are more than just the soma, and the two presentations that we heard before actually confirmed that, right? And so a single patch clamp recording in the somatic region does not allow us to observe other neurites like dendrites or the axon or the axonal initial segment, which might be important and are important for the physiology of these neurons. So what can we do differently? Actually, some researchers, in particularly Itai Hay in 2011 and also others, have used multiple simultaneous patch recording in order to build these biophysical models. In this case, for example, they had 3 different patch pipettes, 1 in the soma and 2 in the apical dendrite. And they used recordings from all these 3 regions to fit the multi-compartment model. And they were able to reproduce very complex dendritic behavior like calcium spikes. And these behaviors are actually quite important for dendritic integration and synaptic plasticity. However, I don't know if you have ever tried to do patch clamping yourself. Even with 1 pipette, that's extremely hard. And doing it with 3 pipettes simultaneously, it's very experimentally challenging. So we propose an alternative method. So instead of having multiple pipettes, we propose to use a single whole cell patch clamp recording from the soma, but to have the neurons sitting on these high density multi-electrode arrays that we develop in the lab. These are super high dense, so the pitch between electrodes is around 15 micrometers. So we can really observe the extracellular activity of neurons at a very fine scale. I would like now to kind of review what's the usual pipeline that it's used to build automatically these kind of models. So we start from the neuron, and as I said before, we have our patch pipette and we extract some data in response to some protocols. Usually step protocols are used of different amplitudes. And then from the same neuron after the experiment, we can finally reconstruct the 3D morphology. Then we need some other ingredients. From the data, instead of using the raw data, we usually extract some features that are a compressed representation of this data. And features can include the number of spikes in the response or the average interspike interval, as well as spike shape features like the amplitude of the action potential or the width of the action potential. And also we need to define the mechanisms of the neuron. And these mechanisms are conductance-based models. And we have to define which ion channels are part of which neuronal sections and how they're distributed. And then the goal of this optimization of this process is to find the parameters of these mechanisms that basically can reproduce the data that we record from. Then we put everything together and we apply the same protocols. We combine them with the morphology and the mechanism information to make a computer simulation of the neuron. And then from this simulation, we extract the same features that we extracted from the data. And we basically try to find the parameters that better fit the feature extracted from the data. In this case, we need to optimize some objective function, and it is usually done via evolutionary strategies or genetic algorithms. And this is the pipeline that Blue PyOpt, which is a software developed by the Blue Brain project, uses. So basically, these objectives tell us how well these features extracted from the simulated data fit the feature extracted from the real data. And then once this optimization is done, we obtain our model, which is the best fit. However, in order to use also extracellular signals, we need to do some modifications to this pipeline. Of course, we need to extend our data acquisition strategy, also including extracellular signals. We need to also extract features from those extracellular recordings. And most importantly, we need to change the simulation in order to also simulate extracellular signals. Regarding the simulations, originally Blue PyOpt support the neuron simulator, but we extended it in order to use LFPy, which is a wrapper to the neuron simulation, which also allows to compute extracellular signals. So the basic idea is that you use neuron to solve the cable equation, and this will give you all the currents that flow in and out of the neuron from all of its compartments. And then using volume conduction theory, we can sum up the contribution of this transmembrane current to compute the extracellular potential at our electrode positions. Then we need to define other features for our extracellular signals. And this is an example of the neuron sitting on top of our simulated multi-electrode array. And here I'm zooming in on the channel with the largest amplitude, and we define some shape features for all of these channels. So we have features like the peak to valley duration, the spike half width, the peak to trough ratio, and so these are defined channel-wise. We have another set of 4 features, which instead are defined with respect to the channel with the largest amplitude. And an example of this feature is the duration, or how much time there is between the negative peak in one channel and the negative peak in the channel with the maximum duration. So these kind of features also allow us to basically describe propagation properties of this signal. And differently from the intracellular features, in this case, for each feature, we have an array of values. So we have these feature maps. So we have to use a different kind of objective function. And in this case, we use the cosine distance between the simulated feature and the experimental feature. Okay, actually, we don't have data yet, but we built some ground truth simulations in order to test this approach. So we started from this layer 5 pyramidal cell model, which was published in 2016 in the Blue PyOpt paper. And then we sampled the parameter space, and we basically built 5 different new models, which we treat as ground truth, and we use them for evaluation. And then we also want to compare 3 different feature sets with the access to different sets of signals. So in the first feature set, basically this is the gold standard of building these models. So we only have access to the somatic traits, and we have 3 protocols, 3 steps with different sizes, and a total of 30 features that we want to optimize. Then we have this multiple feature set in which we have 3 different patch pipettes that can record simultaneously. In this case, we add a new protocol, which is a strong depolarizing pulse, which also allows to basically excite the dendrite in its active regime. And in this case, we have 35 features and 3 access points. And then we have this extra feature set in which, again, we combine the somatic recording with extracellular features. And we have 7 extracellular features, so we have 37 features in total. I will show just some qualitative results. We're still working on doing this in a more systematic way. But in general, what you can see here is the black trace is the ground truth response of the model. And these are the responses to the 5 different test models to 3 different steps of increasing amplitudes. And in all cases, we see that basically the somatic traces are well fitted by all 3 feature sets, even because most of the features that we used are extracted from the somatic traces. But then things get interesting when we start looking at the dendritic response. And these are the responses of basically the Soma and the 2 dendritic sites, which are only available to these orange signals, so the one that use multiple patch pipettes for optimization. And we can see that, in fact, the orange signal fits well the ground truth data in all of the 5 cases. The blue line instead is the somatic feature set, so the one that only has access to the somatic trace, and they can fit well these dendritic responses only in 1 case out of 5, while the extracellular one does somewhat in between, so 3 times out of 5. So in this case, and this case, it can well predict at least the amplitude and kind of the long decay of the dendritic signal. And then, so basically with this extracellular feature set, we can do better than the somatic one, but a bit worse than the multiple feature set. Then finally, I talked about extracellular signals. I just wanted to show you how they look and how the different feature sets fit these kind of signals. So in this case, we use the linear probe that basically traverse the entire neural z-axis. And these are 2 examples we can see, and these signals are actually normalized by their maximum. Otherwise, the dendritic signals would be quite low and hard to see. But we can see that in general, the green and the orange line fit very well the black one, while the blue line sometimes it's a bit worse. And also we basically summarize this by computing for each of these test models, the cosine distance between all of these ground truth signals and the signals, the basically simulated signals using the 3 different feature sets. And we find that the extracellular feature set does almost as well as the one with multiple patch pipettes, while the somatic signal is definitely worse in estimating correctly these extracellular action potentials. So finally, this is preliminary data and only in silico, but these in silico simulations can actually give us some insights also in designing new experiments. And we're still trying to solve some issues. So I showed results only using this linear probe, and ideally we would want to go to something like this, so having thousands of electrodes recorded simultaneously. The problem is that when we go from here to here, simulations start to be quite bulky, and so we're trying to speed up these simulations. Secondly, we use these layer 5 pyramidal cells from the Blue PyOpt paper. However, the basically dynamics of the dendritic trees is quite limited. So we are now in the process of moving towards this Hay model in which there's a lot of richness in the dynamic of the dendrites. And we would also like to change the model to include also a detailed model of the axonal initial segment, which is well known to strongly influence the extracellular signals, and it's also involved in cellular processes like homeostatic plasticity. And then we are currently setting up the experiments, and there are some challenges that we are facing. So first of all, the neurons, they need to be sitting very close to the electrodes in order to get a large signal. So we are starting with cell cultures, which are mainly planar, and they nicely sit on top of the MEAs. And then we're moving towards 2 directions, either going to very thin slices or using organotypic slices, which are slices that are basically cultured on the chip and they become flatter over time. And then since we want to compare the experimentally recorded data, extracellular signals with the simulated one, we need to match the transfer function of these electrodes. So these electrodes are actually physical entities. They have an internal capacitance and resistance. And so we are measuring this transfer function, and we plan to apply it to the simulations to basically compare apples and apples. And finally, I would like to thank all the contributors to this ongoing project, and the ETH and EPFL for the support. And I would like to thank you all for the attention. And let's see if there are any questions.
Thank you very much, Alessio. Do you have any questions from the panelists, maybe? No, not yet.
I guess I have a question.
Oh, okay, go ahead.
So there's a paper from Christoph Koch's lab talking about high amplitude positive spikes. Are you familiar with that? And do you have any evidence of that?
You mean on the dendritic trees?
Yeah, when they recorded out in the dendritic trees, they saw
extracellularly.
Yeah.
I mean, you do see these positive spikes, but I would say that mainly it's kind of hard to distinguish whether they are generated by the intrinsic dendritic conductances, or they are just the effect of this balancing that you see between the current sinks and sources, right?
Right.
So when you have an action potential with strong currents, like inflowing currents in the soma, you would have other like current sources distributed over the dendrite. So you usually see, and we can also see it actually here. So here I'm showing the somatic extracellular action potential, and this is a dendritic one, and you can see that it's kind of reversed, right? Yeah. So I would say that what we measure extracellular is mainly basically the contribution of this distribution of currents, but of course, especially later during the spike, or if you also kind of recruit these active conductances in the dendrites, the shape of these extracellular potentials will be also influenced by these dynamics. And so that's what we hope, that this would be enough to characterize those conductances.
Thanks.
Okay, we have to move on from this room because there's going to be next seminar session here. I want to thank all the speakers for the super interesting talks, and just wish you luck in all your research in the future, and hope to see you around at the conferences.
Thank you.
Have a nice day.
Bye-bye.